Assalamu alaikum dear students. Welcome to Dr. Roomi lectures. Today we are going to study a very important topic from blood physiology, and this is erythropoiesis. You will study what is meant by erythropoiesis, what are the sites where new red blood cells are produced, what are the stages for red blood cell formation, and in the end I will give you a very good mnemonic to memorize the stages of red blood cell production. First, let's see why it is essential to produce new red blood cells. Red blood cells are very important vehicle for transport of oxygen in our blood. There are about 25 to 30 trillions red blood cells in our body. And red blood cells, because they do not have nucleus and organelles, they are filled with a lot of hemoglobin and they have to pay a price for that. And the price is that they are short lived. Their life is just for 4 months, 120 days, as compared to the nerve cells and muscle cells, which live for whole life of a person. Red blood cells live for only 4 months, and this is because red blood cells do not contain DNA, no RNA, no ribosomes. So that's why they are not able to synthesize new proteins. Hence, growth, repair, and the supply of new enzymes is not available. So the aged red blood cells, when passed through narrow and tortuous capillaries of spleen, they squeeze through and they are likely to rupture. Spleen, which is an organ located on the left upper side of our abdomen, is the graveyard of red blood cells, which means spleen will cause death of fragile old red blood cells. This is one of the functions of spleen. Some other functions which are performed by spleen are to store red blood cells in the red pulp. Spleen also acts as a reservoir for platelets, and spleen is also a site where lymphocytes are present, B and T lymphocytes, and they are important to give us immunity. Almost 1% of red blood cells are destroyed daily, and the same proportion of red blood cells must be produced in order to maintain a balance between the destruction and new production of red blood cells. The new red blood cells are synthesized in bone marrow. If there is not a balance between the production and destruction of red blood cells, then the red blood cell number may change in our blood. But it's very important to note that the count of red blood cells is very tightly regulated. Let's suppose there is increased destruction of red blood cells than the production, as happens in case of sickle cell anemia, thalassemia, then the person will end up with anemia. And also, if the formation of red blood cells is relatively less, then once again the person will have anemia, as happens in case of aplastic anemia. Now let's talk about the sites where red blood cells are produced in intrauterine and the extrauterine life. There are three important sites where red blood cells are produced. Number one is the yolk sac, where in the early weeks of embryonic life, red blood cells are produced, and these red blood cells are primitive and nucleated cells. This stage is called as mesoblastic stage. Second stage is hepatic stage, in which during second trimester of pregnancy, red blood cells are predominantly produced in liver. However, a considerable number of red blood cells are also produced in lymph nodes and spleen. In the last months of pregnancy and also after birth, the red blood cells are exclusively produced in bone marrow, and this is called as myeloid stage of red blood cell production. It's important to study these stages of red blood cell production because in some diseases like in thalassemias, leukemias, and some lymphomas, the red blood cells can be produced in liver, in spleen, and this is called as extramedullary erythropoiesis or extramedullary hematopoiesis. In children till the age of 5, all bones in our body are able to produce red blood cells. It's because till age 5 all the bones are filled with red bone marrow. However, from age 5 onwards till age 20, The bone marrow in long bones like in femur, in tibia, humerus is replaced with fatty bone marrow which is not able to produce new cells except at few places at the proximal end of humerus and the proximal end of tibia. So we can say that after age 20 only the membranous bones are able to produce new red blood cells, and the examples of these bones are vertebrae, sternum or breastbone, ribs and the pelvic bone, and these are the sites from where bone marrow may be extracted for examination under the microscope or for bone marrow transplant. Now let's talk about the cells from which red blood cells are produced. In our bone marrow, there is a stem cell which is called as pluripotent or pluripotential hematopoietic stem cell. This is a single type of stem cell from which all the types of blood cells are produced. Red blood cells, white blood cells and platelets. This pluripotent hematopoietic stem cell, it keeps on dividing in our red bone marrow to produce more cells of the same type so that it maintains its number. However, with age its number decreases, and it also divides to produce other types of cells which may become committed to produce a particular line of blood cells. These committed cells, when are grown in culture medium, produce colonies of a certain type. For example, this cell, it is called as colony forming unit spleen. This cell, this is lymphoid stem cell. When this colony forming unit spleen, it further differentiate, it forms colony forming unit blast and colony forming unit erythrocyte. These CFUE, colony forming unit erythrocyte, it further differentiates to make red blood cells. The colony forming unit granulocyte monocyte, it further differentiates and passes through various stages of division and differentiation to make granulocytes, which are neutrophils, basophils and eosinophils, and monocytes, which when enter into blood and then enter into the tissues is matured to become macrophage. Colony forming unit megakaryocyte further differentiates to make the megakaryocyte, which is largest cell in our bone marrow, and this megakaryocyte, it buds off to produce platelets. So in our bone marrow, different committed cells or colony forming cells are present at different stages along with the stem cells which are called as pluripotent hematopoietic stem cells. In order to increase the growth and reproduction of these committed stem cells, we need growth inducers. Some important growth inducers are interleukin 3, interleukin 6 and interleukin 11. These interleukins are proteins and are also called as cytokines. Interleukin 3 is an important growth inducer which affects the growth and reproduction of almost all types of colony forming cells. Differentiation inducers are those factors or proteins which are involved in causing differentiation of these cells in a certain line. So they cause these cells to move one or two steps forward towards the adult stage of these cells. The differentiation inducers do not cause the growth of a specific type of cells, rather they move the cells one or two stage forwards towards the adult final stage of the cells. The formation of growth inducers and differentiation inducers is because of some factors which are outside of the bone marrow. For example, when our body is exposed to low oxygen level or hypoxia, there is production of erythropoietin, which is a very important factor that controls the production of red blood cells. And also when we are having infection in our body, then there is production of some kind of cytokines like interleukins, which are responsible for increasing the growth and differentiation of various types of white blood cells. Best studied mechanism that controls the growth and differentiation of red blood cell production is release of erythropoietin hormone from kidneys in response to hypoxia. Erythropoietin hormone acts on bone marrow to increase the production of red blood cells. We'll study this mechanism later on in another session. Now let's talk about the stages of erythropoiesis. First, about this term erythropoiesis. This means formation or production of new red blood cells. When you use this term hematopoiesis or hemopoiesis, that means formation of all the types of blood cells. But erythropoiesis is specifically for formation of new red blood cells. The first stage is proerythroblast, also called as megaloblast. This is a large cell which is the very first cell produced from the clone-forming unit, the erythrocyte, and this cell has the features of red blood cells. This is the first cell in which synthesis of hemoglobin begins. Its size is about 20 micrometer. It has a large nucleus and the chromatin material in the nucleus is diffuse, which means this is a very active cell. The cytoplasm is basophilic because it's having a lot of ribosomes which are involved in making proteins. So it's an active cell, and those ribosomes, because they have acid, ribonucleic acid, and acid stains with basic dyes. So that's why we can say that its cytoplasm will stain with basic dyes. So it's basophilic. And also, two or more than two nucleoli may be present in the nucleus, which also is a sign of activity. So it's an active cell. Proerythroblast, it divides for several times to make the next type of cell in this series, which is called as basophilic or basophil erythroblast. This cell is also having a cytoplasm which stains with basic dyes, hence the name basophil erythroblast. It's also called as early normoblast. Its diameter of the cell is slightly smaller than the proerythroblast. It's 15 micrometer. Now in this case, the nucleoli disappear in the nucleus and the chromatin material in the nucleus gradually condenses. It becomes less diffuse are more condensed. Next stage is polychromatophil erythroblast. Here the word poly means this stains with two types of dyes, basophilic and acidophilic dyes. Basophilic because its cytoplasm is still having a lot of ribosomes which are acidic and they stain with the basic dye, and because now hemoglobin content is gradually increasing and that starts appearing. Hemoglobin synthesis began in the proerythroblast, and now the hemoglobin is enough in concentration that it starts appearing. That's why hemoglobin, which is a basic protein, stains with the acidic dyes. So that's why it also gives a little bit pink color in the cytoplasm. So both bluish and pink stain are taken by the polychromatophil or polychromatic erythroblast, which is also called as intermediate normoblast. Its diameter is smaller than the basophil erythroblast, and this is just 10 to 12 micrometer. Nucleus further condenses, and the next stage is orthochromatic erythroblast. Now in this case, enough hemoglobin has been accumulated in the cell that the cytoplasm stains pink now with the acidophilic dyes. This is also called as late normoblast. Its diameter is 8 to 10 micrometer. Now the nucleus has further condensed and it has become very small in size. It is so darkly stained and small nucleus that it is also called as ink spot nucleus. This nucleus will gradually disintegrate and will disappear from the cell, and this process is called as pycnosis. Now the next cell in this series is reticulocyte. This cell is also called as immature red blood cell. Its diameter is slightly larger than the mature red blood cell. It's about 8 micrometer as compared to the mature red blood cell, which is about 7.8 micrometer in diameter. We call it reticulocyte because our reticulum, our reticular network of the remnants of ribosomes, mitochondria, and Golgi apparatus is still present inside the cytoplasm of the cell. Otherwise, the nucleus has disappeared. And these remnants of organelles are stained with supravital dyes. The remnants of organelles are the reticulum disappears from reticulocytes within 1 to 2 days. And because these are short-lived cells in our blood circulation, so that's why their percentage is very small in our blood. They are from 0.5 to 1.5%. On average, they are about 1% in our blood circulation. Reticulocytes enter from our bone marrow, passing through the gaps of sinusoidal or discontinuous capillaries into our blood circulation. And when these reticulocytes they have entered into our blood circulation, they do not come out when they are passing through various capillaries throughout the body, because those capillaries they are small in diameter, and also they do not have large gaps as are present in the sinusoidal capillaries of the bone marrow. We have just studied that reticulocytes are 1%, less than 1% in our normal circulation. However, the percentage of reticulocytes can increase in those conditions when there is rapid erythropoiesis going on in our bone marrow. For example, in case of hemorrhage, when someone has lost blood, in response to that, our bone marrow may try to increase the production of red blood cells, and that rapid proliferation of red blood cells in the bone marrow may increase the release of immature cells, reticulocytes, into the circulation. And also in the case of some anemias, when there is increase in destruction of red blood cells, in response to that, the bone marrow is trying to increase the production of red blood cells, and then even the immature red blood cells will enter into our circulation. And also, if in some case a person is deficient in some vital nutrients like iron, folic acid, B12, if the person is given that deficient nutrient, then in response to that, the bone marrow will increase the production of red blood cells, and then the production of reticulocyte can increase and its percentage can increase, which shows a response of the bone marrow to that deficient nutrient. And also, if a person is given erythropoietin hormone, which stimulates the production of red blood cells from the bone marrow, that will also increase the production of red blood cells, and one manifestation of that will be the reticulocyte percentage will increase in our circulation. So in some conditions where red blood cell production is very rapid in our bone marrow, then the percentage of reticulocyte can increase up to 30% or even more in such conditions. And the final stage in erythropoiesis is erythrocyte, or the mature red blood cell. Its diameter is like 7.8 micrometer. It has no nucleus, no mitochondria, no Golgi apparatus, no ribosomes, and no endoplasmic reticulum. And this is because we want to fill this red blood cell with a lot of hemoglobin so that it can carry a lot of oxygen. And when this red blood cell is fully loaded with hemoglobin, then in the packed blood, the content of hemoglobin is 34 grams per deciliter. I'm not talking about the full blood. I'm talking about the packed blood cells. It has a content of 34 grams of hemoglobin per deciliter of packed cells, which is the maximum limit up to which we can pack hemoglobin in these cells. And now, a very interesting mnemonic to memorize the stages of erythropoiesis. And the mnemonic is: profitable businesses pollute our rural environment. P in the profitable is for proerythroblast, businesses B, basophil erythroblast, pollute, polychromatophil erythroblast, our orthochromatic erythroblast, rural for reticulocyte, and environment for erythrocyte. Using this mnemonic, you can memorize the 6 stages of erythropoiesis. I hope you must have enjoyed this session. In our next session, we'll talk about a very important regulator of erythropoiesis, which is erythropoietin hormone. We'll talk about its stimuli and the site from where it is produced, what are its actions on the bone marrow. We'll also talk about the biotechnology which is producing erythropoietin and its clinical uses, and we'll also give a touch of doping, how and why some athletes use erythropoietin, and what are legal and unethical aspects of using erythropoietin for athletic performance. Thank you so much for watching this video. See you next time with another video.